DNA Extraction Lab, Biology 2, Spring 2020, Miss McCoy. What is DNA and what does it do? Deoxyribonucleic acid, DNA, is a molecule present in all living things, including bacteria, plants, and animals. DNA carries genetic information that is inherited or passed down from parents to offspring. It is responsible for determining a person's hair, eye, and skin color, facial features, complexion, height, blood type, and just about everything else that makes an individual unique. But it also contains all the information about your body that is the same in all human beings. In other words, your DNA is like a blueprint for your entire physical growth and development. Your DNA blueprint is a combination of half of your mother's and half of your father's DNA, which is why you have some features from each parent. DNA contains 4 chemical units, referred to by the first letter in their names: A, adenine, G, guanine, T, thymine, and C, cytosine. These 4 DNA letters make up a code for genetic information. The letters of the DNA code are similar to the letters of our alphabet. The 26 letters of our English alphabet spell words, which can be arranged in infinite ways to create messages and information. Similarly, the 4 chemical letters of DNA are organized to make messages called genes that can be understood by cells. These genes contain the information to make proteins, which are responsible for almost all of your body's structures and functions. A gene is like a recipe, since it contains all the information needed to make a protein. Your DNA sequence is the particular arrangement or order of the chemical letters in your complete DNA collection, or genome. Scientists have determined that human DNA sequences are 99.9% identical. It is that the 0.1% sequence variation from person to person that makes each of us unique. In other words, what makes you different from your classmate is an occasional difference in a letter in your genome. Where is DNA found? The basic units of an organism's body are cells. They make up all your tissues and organs: muscles, brain, digestive system, skin, glands. Cells are compartments with membranes made from protein and lipids that keep them separate from other cells. Within the cells are further compartments with specialized functions. One compartment, called the nucleus, is like the cell's control headquarters and contains the DNA molecules, which are the master instructions for the functions of the cell. The DNA is organized into 46 tightly coiled structures called chromosomes. Actually, it is more accurate to say that there are 23 pairs of chromosomes in each cell, 23 from your mother and a matching set of 23 from your father. What does DNA look like? At the molecular level, DNA looks like a twisted ladder or a spiral staircase. The ladder actually contains 2 strands of DNA with pairs of the chemical letters A, G, T, and C forming the rungs. This structure is called a DNA double helix because of the spiral or helical form made by the 2 DNA strands. Each strand of DNA is very long and thin and is coiled very tightly to make it fit into the cell's nucleus. If all 46 human chromosomes from a cell were uncoiled and placed end to end, they would make a string of DNA that is 2 meters long and only 2 nanometers, yes, that's 2 billionth of a meter wide. How can we make DNA visible? DNA is only about 50 trillionth of an inch long. The reason it can be seen in this activity is because students are releasing DNA from a number of cells. This happens when the detergent or dishwashing liquid breaks or lyses the membranes around the cell and the nucleus. Once released, the DNA from both broken open cells intertwine with the DNA released from other cells. Eventually, enough DNA intertwines and becomes visible to the eye as whitish strands. One strand of DNA is so thin they would never be able to be seen without using a microscope. Step 1. Collect cells. To see your DNA, you will collect cells, break them open, and condense the DNA from all the cells together. You can collect thousands of cells from the inside of your mouth just by gargling with saltwater solution. This will dislodge the cells. The type of cells that line your mouth divide very often, coming off very easily as new cells replace them continuously. Step 2. Break open, lyse, the cells. Once you have collected your cells, the cells need to be broken open to release the DNA. Detergent will dissolve the membranes of your cells, just like dishwashing detergent dissolves fats and proteins from greasy pans, because cell membranes and nuclear membranes are composed of fats and proteins. Dissolving the membranes results in the release of the DNA. The process of breaking down the cells is called lysis, and the solution containing the detergent is called lysis buffer. Step 3 and 4 condense the DNA. Strands of DNA are so thin it's not possible to see them when they are dissolved in solution. Think of a long, thin strand of DNA as fine white thread. If one piece of thread were stretched across the room, it would be difficult to see. To make the thread more visible, you could collect it all together and pile it on the floor. In this laboratory experiment, you will use cold alcohol to bring the DNA out of solution, or precipitate it. Cold alcohol creates a condition in which DNA doesn't stay in solution, so the DNA clumps together and becomes a solid mass that you can see. Lab procedures. At your station is the following items: a test tube rack with 3 to 4 clean test tubes, 3 to 4 pipettes containing clear dish detergent, 1 graduated cylinder with isopropyl alcohol with blue color, and 3 to 4 Dixie cups. The teacher will come around the room to give each student the needed saltwater solution. Step 1. Gargle for 1 minute with the saltwater solution. Step 2. Spit the contents of your mouth back into the Dixie cup. Step 3. Carefully pour the contents of the cup into your test tube. Step 4. Add the dish soap from the pipette by taking some of the test tube contents into the pipette, then releasing it back into the test tube. Stir gently with the pipette. Step 5. Slowly pour a small amount of alcohol mixture into the test tube. Step 6. Wait 5 minutes. What do you see? Cleanup procedures. Throw away the used Dixie cup and pipette in the regular trash. Then take your test tube to the sink and with water running, pour the contents down the sink. Follow that by rinsing the test tube and placing it in the rack next to the sink. As with any lab procedure, please leave the room clean and tidy.